Okay. Good evening to all of you. Think කොහමද? Okay. Today we are going to do a new and cool lesson. So that's photosynthesis. Okay. Uh, Already you know that photosynthesis. So what happens inside a chloroplast? Okay. So what are the pigments involved in that? So what are the factors affecting to the photosynthesis? So these things are to be discussed. Okay. Let's move. Yes. Live file is ready. YouTube live is running. Okay, I forget that. වෙලාවට run උනේ file එක. මේ වෙලාවේ live සම්බන්ධ වෙන්නත් පුළුවන්. I don't know the quality. quality එක නම් දන්නේ නැහැ. හරි. ඒ කියන්න අමතක වුණා. වෙලාවට auto play එකක් ගියේ නේද? Shanuka and so Sadisha, so you can tell your friends to use YouTube for the streaming, so you can watch. Password link yavanna, so you can log into the Zoom meeting directly if you want to question for direct questions, and if you want to just see the process what happens here, so you can log into YouTube by using your Gmail. Okay, it's not members only, so you can freely watch that. Okay, so today photosynthesis. Okay, so what does it mean by the term photo? Okay, photo means light. And synthesis, what does it mean by synthesis? Synthesis means, yes, produce. Photo, so that's light. Okay, synthesis, produce. So producing of foods by the green plants using the energy of sunlight. So that whole process is known as the photosynthesis. Okay, if we graphically illustrate this, if you graphically illustrate that in your childhood, okay, when you are in grade 6, you learned it like this, a little plant, that's what you drew. Okay, that root system. Okay, so tap root system. Good. So what are the factors affecting to the photosynthesis process? Okay, when you learn in grade 11 tissues lesson, okay, you come across plant tissues. Okay, so and we know what are the layers of tissues in this plant leaf. So that's a modified parenchyma tissue. ඔගොල්ලෝ ඉගෙන ගත්ත විදිහට modified parenchyma තමයි තිබ්බේ. So what are they? So this modified parenchyma cell is epidermis, and you observe another cell layer in the cross section of this plant leaf. So that's this palisade mesopyle. Mesopyle means parenchyma. Okay, so these cells are existing inside that, and you observe this. What's this? Spongy mesopyle cells. Spongy mesopyles. Okay, large central vacuole, so that's characteristic feature. So that lower epidermis you could observe lower epidermis, epidermal cell layer, and here's spongy mesopils. So you can observe lots of intercellular spaces here, and this palisade mesopil cell somewhat elongated. Okay, elongated vacuole, nucleus exists. All the instances, nucleus. Okay, so this lower epidermis, specialty of this, all the cell layers of the cross section of the plant leaf. So these cells are having chloroplasts. Okay, so these are chloroplasts. If I draw that clearly, you learned that in grade 10. Chloroplast in this way, a huge organelle. Okay, and it is double membranal organelle, double membranal. So this membrane make another set a container like structures. We call them granum or thylakoids. Okay, so chlorophyll pigments exist here. That green color is not contrasting. Okay, so green color pigments exist on this chloroplast. Chloroplast. So these are the chloroplasts in plant cells. So as you have already learned this lesson, tissues, you know, so this palisade or spongy mesophyll cells are having chloroplasts. Okay, and inside the chloroplasts, you can observe chlorophyll pigments. Chlorophyll pigments. Okay, so what are the factors affecting to the process of photosynthesis? Okay, you know already, the photosynthesis means the process of producing foods required by the plants and all the organisms living in this world. Okay, so that required food is produced by using a special process. That process is known as photosynthesis. Photo means light, and synthesis means, okay, production. So there are different types of uh, nutrition habits. One is autotropism, as you learned in grade 6. Okay, autotropism. Autotropism means, so they can produce their own foods themselves, just like green plants. So they are producing their own foods by using the energy of sunlight. Okay, so whatever the type of light with green, so with red and blue. Okay, if there's red and blue color rays, so they can um, produce foods by using the energy of that red light and blue light. Okay, the light ray type that is not used for the photosynthesis is green. That's why it is emitted. That emitted color can be seen as a green color. That's why these chloroplasts and plant leaves are with that typical color of green. Okay, so light energy is the main source. There are some types of bacterium especially, so they are not using this light energy for the production of energy. That means for the production of foods. So in that case, that also a type of autotropism. They can produce their own foods, but not using the energy of light. So they are using the chemical energy. Okay. By using the chemical energy, they can produce their own foods. In that case, so that autotropism is known as chemoautotropism. Okay, chemoautotropism. Chemoautotropism. Okay, so now what we are going to study is photoautotropism, because they are using light energy instead of chemical energy. So therefore, so what we are going to study today is phototropism. Okay, let's see what are the things in your tute. Okay, now I'm going to share that. Don't touch your screen. I saw several students touch your screen. So you are not touching and writing on your screen, but also all of us can see that. Okay. Um, let's do it again. Okay. So this is the front page of Chartute. Yes. Yes. This is what you learned last week, I think. So this is the plasma membrane. I have described you several times what this plasma membrane is. Okay. This is how your tute is. I have already sent it to your WhatsApp groups. Yes, look at this. All the living organisms are depending on the foods produced by green plants. Okay, actually, you have to know not only the green plants, but also there are some other types of organisms who produce their own foods themselves. Their own foods themselves. One example is a special type of bacteria. We call that cyanobacteria. So another type of microorganism, algae. So that's also involved in this process of photosynthesis. So, but here, the large amount of food is produced. So involvement in large scale, involvement is with these green plants. So organisms show two major types of nutrition. So what's that? Autotropism. Okay. Next one, heterotropism. Okay, let's begin the lesson from here. Okay, take down the topic photosynthesis. Okay. And that take down photosynthesis is photosynthesis is the process of the process of producing foods producing foods. Okay, in science we use for the production of new thing as synthesis. Okay, uh, process of producing foods within bracket take down synthesizing of foods or synthesis of foods. Synthesis, production means synthesizing, a kind of production. Another term we use for that is synthesis. Okay, from the beginning, the photosynthesis is the process of producing foods by using the energy of sunlight. By using the energy of sunlight. Okay, we are going to do a certain correction here. Here we have used in the tute produced by green plants. No matter, that's correct. But you have to know there are other types of organisms involved in this process. Therefore, photosynthesis is the process of producing foods by using the energy of sunlight and take down by the photoautotropic organisms. Okay, by the photoautotropic organisms. Okay, next point. So this lesson comes under nutrition. Next point, take down. Mainly there are two types of nutritions. Mainly there are two types of nutritions. Mainly there are two types of nutritions. First one, autotropism. Okay, first type is autotropism. Okay, now autotropism. So you have to know there are two types of autotropism. Okay, so let's describe that later. Autotropism. Next one is heterotropism. Heterotropism. Okay, next point, take down. Yes, you can see that here. Autotrophic organisms are the organisms. Autotrophic organisms are the organisms that produce their own foods themselves. Autotrophic organisms are the organisms that produce their own foods themselves. Okay, as I mentioned to you last session, so this lesson is not to study biology. Okay, so these things you know. So you have to know how to explain, how to describe experiments. Okay, that scientific writing you can learn here. Okay, I think uh, so. You have to know the way of writing a scientific report. So that part is learned from here. Okay, good. So autotrophic organisms are the organisms that produce their own foods themselves. Their own කියන part එක ටියුට් එකේ නැහැ. ඔවුන්ගේම ආහාර. Okay. There are several types of autotrophic organisms as I explained to you earlier. So autotropism is of two types. Next, take down, list them down. Proto-autotrophic. It's adjective. Proto-autotrophic. Okay. As a noun, you can write proto-autotropism. Okay. Proto-autotrophic organisms or proto-autotropism. Okay. So you have to describe that. Use light energy as the energy source. Yes. For the production of energy, light energy is used by these organisms. As the lights, as the energy source, they use light. Therefore, they are known as proto. Proto means light. Okay. Next type, chemoautotropism or chemoautotrophs or chemoautotropic organisms. Rasa සංශ්ලේෂකයෝ කියලා සිංහලෙන් මේකට කියනවා. Yes, they use chemical energy as the energy source. By using the energy source, chemical energy, they produce their own foods. Therefore, as they are producing their own foods, they becomes autotrophic organisms. As they are using the energy of chemical reactions, they are known as chemoautotrophs. Okay, good. So these are the things that overall process graphically and all the things there. So you can read that later. Yes, green plants are considered to be autotrophic organisms. Briefly explain on this. Before that, we have to know, so what are the types of organisms who involve in the process of photosynthesis? මේ photosynthesis කියන ක්‍රියාවලිය පෙන්වන ජීවීන් කවුද කියන එක පොඩ්ඩක් පැහැදිලි කරගන්න ඕන. So we have to know that. Okay. I'll switch off this screen share for a while. Okay. Yes, that's a question. පුතෙක් ප්‍රශ්නයක් අහනවා. Sir, in the photosynthesis, can they use light energy from other sources except sun? Yes, that's true. So that's what we practice usually in greenhouses. So greenhouse effect is not that. So we use fluorescent bulbs light. Incandescent light source can be used, but more than the production, we have to spend money lot because incandescent light consume much more energy and efficiency is low. Uh, Rather than the light energy, it produces the heat. Think incandescent light, filament bulbs පාවිච්චි කරන්න නැතුව tube lights නැත්නම් inflorescence CFL bulbs පාවිච්චි කරනවා or LED can be used. But most suitable method is incandescent filament bulbs, but it consumes much more energy. So therefore, that CFL are used. Efficiency එක තවත් වැඩි කරන්න පුළුවන් පුතා කොහොමද? You can use blue bulbs, blue light and red lights. අපි white light දුන්නට, okay, I'll show you that next step. Look at this. Hey, Balan. You can see that here. Several experiments. Yes, there's that graph. You can see the performance or absorbance of energy. So that's very high in blue light and red light. So minimum absorption can be seen in that green color. That's why that green color light is emitted by the chloroplasts and chlorophyll pigments. Okay, I think that's clear for you. Thank you for your question. Mama prashna ahanawata kemathi. Okay, now I'm going to stop the share. So these are the things we discussed. Okay, now take down types of organisms involved in the process of photosynthesis. Types of organisms involved in the process of photosynthesis. Okay, first one, green plants. Take down green plants. Next, take down under bacteria, you learned cyanobacteria. Cyanobacteria. Next example, algae. Algae. So what are the examples for algae? Chlamydomonas, Spirogyra, right? Chlamydomonas, Spirogyra, ulva. Yes, Chlamydomonas. Good. Another type of organism who perform this photosynthesis is what? Bacteria. What about fungi? Never. Fungi cannot do this photosynthesis. Bacteria, fungi, algae is okay. Next one, protozoans. What do you think? What are the examples for protozoa? Amoeba, paramecium, දෙන්නම photosynthesis කරන්නේ නැහැ. තව එක්කෙනෙක් ඉන්නවා. ඔයගොල්ලන් හොඳට දාන්න. Yes, Euglena. Take down protozoa, Euglena. Protozoa, yes, that type of organism, microorganism, protozoa. An example is Euglena. All the protozoans cannot produce foods by photosynthesis, but Euglena do that. Euglena. Okay, so these are the basic things. This is the background, what we learn here. මේ අපි ඉගෙන ගන්න වටපිටාව. දැන් සුදුසුයි. Now you are qualified to learn this. Okay, next main topic take down. Factors. කෙනෙක් චුට්ටක් වැඩ පෙන්නනවා මේ අවස්ථාවේ. Cyanobacteria belongs to the domain bacteria කියලා කියනවා. හරි යාගේ ආඩම්බරයි ප්‍රකාශ කරනවා මේ වෙලාවේ. So I'm glad to hear from you. අන්න එහෙමයි ඉගෙන ගන්න ඕනේ. එක්සෑම් එකත් ලියලා හැම ඉන්නේ කට්ටිය ලකුණු හුඟක් අයගේ අඩු වෙලා තියෙනවා. හුඟක් අයගේ වැඩි වෙලා තියෙනවා. හරි? That superiority complex එක ගේමක් නෑ කියලා හිතපු අය ලකුණු 10ක් 15ක් අඩු වෙලා තියෙනවා. හුඟක් අයගේ ලකුණු 20කින් 30කින් වැඩි වෙලා තියෙනවා. So I am happy to say that. හරි? ගැම්මක්. ආන්නේ බය තියාගන්න ඕනේ. තේරුණාද? එතකොට තමයි පුළුවන් වෙන්නේ. සැහැල්ලුවට ගත්තොත් බෑ. Good. Yes, topic is factors affecting to the photosynthesis. Now look at the board. Yes, energy of sunlight. Energy of sunlight. This is the main source of energy as we are discussing this photoautotropism. Sunlight, one factor. Actually, that factor is energy source. For the absorbance of this energy of sunlight, a color pigment is required. So that color pigment is in the cells of the plant leaf. So these cells are a type of parenchyma modified, palisade mesophyll, spongy mesophyll. Okay, palisade mesophyll, spongy mesophyll. You have taken down that. පුතාලා එක ඉගෙනගෙනයි තියෙන්නේ. So that color pigment is chlorophyll. Chlorophyll pigment. Okay. Yes, you can observe here guard cells. If I draw the 3D view of that, so it's like that, guard cells. It's also having these chlorophyll pigments inside the chloroplasts. Usually chlorophyll pigments exist inside the chloroplast. But there are some instances where these chlorophyll pigments just exist in the cytoplasm. One example. Ko බලමු දැනුම. දැන් මේ ටිකකට කලින් හරි මට වැඩ පෙන්නපු වැඩෝ ටිකකෝ බලමු. Usually chlorophyll pigments exist in chloroplasts. Chloroplast කියන organelle එක ඇතුලේ තියෙන chlorophyll. But there are some instances where these chlorophyll pigments exist in the cytoplasm. වැඩෝ වැද්ද වුණා වගේ හරි. වැඩෝ ටික වැද්ද වුණා. Cyanobacteria කිව්වා නේද? Cyanobacteria එකට තියෙන organelle එක තමයි ඇහැට පේන්නේ නැති බැක්ටීරියා වලට තියෙන්නේ. මොකද්ද? Ribosome. Protein synthesizing. Yes. Ribosome. They don't have that chloroplast, but cyanobacteria produce their own foods by the process of photosynthesis. So in that case, their chlorophyll pigments exist in the cytoplasm. පුතා සමහර අවස්ථා තියෙනවා. There are some instances in the world of microorganisms. We can observe huge chloroplasts. One example. Chloroplasts තියෙන තව අවස්ථාවක් හම්බෙනවා. මොකද්ද? Yes, algae. algal chloroplast is the largest chloroplast. එතකොට plants second වෙනවද නිකන් plants එයාලගේ ලොකු නැද්ද? හරි? එහෙම එකක් නැහැ. හරි? එහෙම එකක් නැහැ මේ plants වල. චූටි වෙන්න තමයි එයා වැඩික් වෙන්නේ. So efficiency of the conversion of that production of foods by the process of photosynthesis, that efficiency increases when chloroplast becomes small. Because you know, when small cells, large number of small cells exist rather than a single cell, so their efficiency increases. So likewise, the most efficient chloroplast can be seen in the kingdom of plant, okay, plantae, because small chloroplast, large number of chloroplast, but size is small. In the world of microorganisms, especially algae. So algae chloroplast is very big. For an example, we can take Chlamydomonas. ලොකු chloroplast එකක් තියෙන. ඒක එච්චර දියුණු ලක්ෂණයක් නෙමෙයි. එහෙම හරියට chloroplast එකක් තියෙනවා. But bacteria like cyanobacteria, they don't have even a chloroplast. They have only chlorophyll pigments. හරි? ඒක ලොකු දැනුමක්. හරි? ඔයගොල්ලෝ ඒක ගැන එච්චර අවධානය යොමු කරලා තිබ්බේ නැහැ. Chloroplast and chlorophyll, don't confuse with that. Yes, sunlight, one factor. Another factor is chlorophyll pigments. Next one is absorbed through this guard cell opens, and this opening is known as stomata. Stomata. So this is stomatal opening. So stomatal opening. So this is known as stomata. So through this stomatal opening, carbon dioxide is absorbed. So another important factor is carbon dioxide. So this is the most important process. If not, what happens? Greenhouse effect may occur. So global warming occur. Then liquefying of glaciers and polar ice caps, it starts to liquefy. So what happens? Small islands submerge to seawater. කුඩා දූපත් වැහෙනවා. ලෝකෙ උණුසුම් වෙනවා. මේ ක්‍රියාවලිය වුණ නැත්තම්. So that's another importance of this photosynthesis, absorbance of carbon dioxide from the atmosphere. So what we usually do is again and again, so continuously we add carbon dioxide to the atmosphere. As a result of that, what happens? Greenhouse effect occurs. ලෝකෙ උණුසුම් වෙනවා because this polyatomic carbon dioxide gas can absorb heat. It can absorb heat. Okay? ඉතින් carbon dioxide උරා ගන්න එකම ක්‍රමය තමයි මේ photosynthesis. Another importance of this photosynthesis, but main importance of that is the production of foods, glucose. Okay. So another important thing, in addition to the main food glucose, that C6H12O6, what happens? As a byproduct, oxygen gas is produced. Oxygen gas is produced. Okay, I think if you can add this diagram to your note, that's very advantageous. දැන් අඳින්න වෙලාවක් නැහැනේ. පුතාලා print screen එකක් ගහගන්න. Good. You have already taken a print screen. Okay, so that's chlorophyll, a factor. Another one is sunlight. So next one is carbon dioxide. The most essential, another factor is, yes, it is absorbed from soil. That's H2O. Okay, water, main factor. Okay, now I'm going to erase this part. Okay, let's list down these required factors for the photosynthesis. Factors required for the process of photosynthesis. Okay, first one, sunlight. Actually, that's the energy source. Energy source. Next one, water. Okay, let's write sunlight is absorbed with the help of that chlorophyll pigments. Okay, let's write that chlorophyll, chlorophyll, that pigment. Okay, another factor, carbon dioxide, that's CO2. Another one is H2O. So they are the factors, actually they behave as raw materials. So we can consider this green plant or cyanobacteria or algae. So we can consider them as a factory. Okay, we produce energy. So this carbon dioxide and water, so that's the raw material. Carbon dioxide and water is the raw material. So this is the introduction. එතකොට lesson එකේ වැදගත් කොටස් තියෙන්නේ මොනවද? Experiments. So we have to know them. So they will be appeared in your exam. So lots of your abilities can be tested by using the experiments associated with photosynthesis. Okay, now you can list them down. Factors required for the photosynthesis. Okay, good. Next, we are going to study okay, George is asking a question. So what is the energy that solar energy is converted to? Okay. What's the energy conversion there? What's the type of energy stored in foods? You learned that. That's chemical energy. That's chemical energy. Okay. Me grade 6 ළමෙක්ගෙන් අහුවනම් කියනවා චූටි මල්ලි කෙනෙක්ගෙන්. Okay, let's move. The process of photosynthesis as a chemical equation. The process of photosynthesis as a chemical equation. Okay, so we have to know the word equation as well as the chemical symbol equation. Okay, look at this. First one, word equation. Word equation. Yes. What are the reactants? So you write water absorbed from the root hairs from the soil. Next, water reacts with carbon dioxide. Carbon dioxide. Yes, what is formed? Glucose. You know, glucose is a type of monosaccharide, glucose, fructose, galactose. Okay. So this initial product, as a byproduct, oxygen is also produced. Oxygen, oxygen, water, carbon dioxide react together to form glucose and oxygen. So what is the energy required for that? Yes, sunlight. Yes, energy of sunlight is absorbed with the help of what? Chlorophyll. So this is the word equation. Take it down. Okay, I request all of you to type the letter P. හරි? හැමෝම P කියන letter එක type කරන්න. Because I want to check the people who don't respond and will be removed. Once removed, can never be joined. For what? මොකකටවත් නෙමෙයි. Just you write P to show you obedient. P, okay, not B. Some of you type in B. Okay, thank you. Yes, within several intervals, I usually do these things. Again, in another few minutes, or maybe 20 or 30 minutes, I'll check it again. Okay, so I don't tell the reason behind that. Okay, now let's see. So water, carbon dioxide, so they are the reactants or raw materials. So sunlight is the energy source. Chlorophyll is the pigment used to absorb the energy of sunlight. So glucose is the main product. Oxygen is the byproduct. Okay, next you have to write balanced chemical equation. Balanced chemical equation. Okay, let's write that balanced chemical equation. Water, so that's H2O. Carbon dioxide, so that's CO2. Sunlight, chlorophyll. So no matter, you can write H nu. Planck constant into nu means frequency, but I think you had better to write sunlight. Chlorophyll. Yes. Glucose, you know that C6H12O6, and oxygen O2. Yes, next we have to balance this. So that's very easy. Except glucose molecule, for all the other instances we put the coefficient of 6, then it is balanced. 6 times H2, 12 hydrogen. Okay. 6 times O, 6O, and here 6 times 2 oxygen. So that's 12, 12 with the 6 altogether 18. The same thing happens. 12 plus 6, 18. Carbon, that's 6, already balanced. So this is balanced chemical equation of the photosynthesis. Good. Okay, now I'm going to clean the board. Okay, I'll erase all these things. Okay, next thing we have to learn is we have to prove actually these factors are required. These products are produced. We have to experimentally prove them. මෙන්න මේ දේවල් අවශ්‍යයි කියලා පරීක්ෂණාත්මකව පෙන්වන්න සිද්ධ වෙනවා. So we have to show it. Okay. But there's a problem. So how to show? කොහොමද පුතා අපි මේක පෙන්වන්නේ මේවා ඕන කියලා? For instance, the requirement of water. ඒක නම් පෙන්වන්න හම්බ වෙන්නේ නැහැ. වතුර ඕන කියලා පෙන්වන්න හම්බ වෙන්නේ නැහැ. මොකද if we control water, plants gets died. ගහම මැරෙනවා. ඉතින් අපි හිතා ගන්නවා photosynthesis කරන්න ගහක් තියෙන්න ඕනනේ. There must be a plant, living organism. Therefore, water is required. ඒක ටෙස්ට් කරලා පෙන්නන්න බෑ. හරි? ටෙස්ට් කරන්න වතුර නොදී බලන්න ඕනේ photosynthesis වෙනවද කියලා. But if you control the supply of water, plant මැරෙනවා. Next, take down the topic. How to prove that above factors are required for the photosynthesis. So that's a kind of question, but that question is actually a topic. That question is actually a topic. Hari. Then putala ඔක්කොම, now you have to keep your pen aside. පුළුවන් නම් ෆෝන් එකත් steadily තියාගන්න ඉස්සරහින් තියාගෙන හොඳට අහගෙන ඉන්න. Okay, now I'm going to explain this. So this is 100% logical because this is biology. Okay, so there's a logical part. මේකේ පාඩම් කරන්න කිසිම දෙයක් නැහැ. Nothing to be cramped. The only thing you have to do is listen contemplately and think logically. So that's the thing you have to do. Okay, look at this. How do you prove that photosynthesis has taken place? Okay, so before doing the experiment for the requirement of each of the factors for the photosynthesis, we have to know that photosynthesis has taken place. Okay, so under that, next subtopic is before. Don't write. Okay, so we write the next subtopic after writing this little statement. Before testing the requirement of the factors, we have to find a method to prove that photosynthesis has taken place. මේ ඔය කියන වතුර ඕනද? ඒක බෑ කිව්වනේ. කාබන් ඩයොක්සයිඩ් ඇත්තටම ඕනද? සන්ලයිට් ඕනෙමද? මේ photosynthesis එක. මෙන්න මේවා ටෙස්ට් කරන්න කලින් එව්වා දුන්නොත් photosynthesis වුණා කියලා දැනගන්නේ කොහොමද? How do we identify that photosynthesis has taken place? So before that, we have to do a test to confirm that photosynthesis has taken place. Okay, before testing the requirements of the factors, we have to find a method to prove that photosynthesis has taken place. Okay, photosynthesis has taken place. So what we are going to prove, we are going to prove that photosynthesis has taken place. The first thing we have to do. Okay, so we give a certain factor. So photosynthesis takes place. Photosynthesis occurs. In that case, we know that factor is required for the photosynthesis, but we cannot confirm it. How to confirm? We control that factor. We don't give that factor to that plant. Okay, then we can observe that plant is not doing photosynthesis. Then we can conclude. Our conclusion is that factor is essential. Because of the absence of that factor, no photosynthesis. Therefore, but when we want to jump to that conclusion, we have to know a method how to prove that photosynthesis has taken place. Yes, how to prove? Now I need your answer, your suggestions. How to prove that photosynthesis has taken place? Yes, if photosynthesis has taken place, definitely glucose C6H12O6 is produced. But we don't test this glucose because glucose is the initial product. Okay, and oxygen is the byproduct. So this is the overall process. 6H2O plus 6CO2. So with the sunlight, H nu, and okay, next chlorophyll. It's an overall process. If photosynthesis happens, if photosynthesis occurs, so these glucose molecules are produced. But we are not going to test this, the existence of glucose, because this is the initial product. Okay, so as this is initial product, what happens later? large number of glucose molecules polymerize together. Large number of glucose molecules polymerize together to form what? Starch. Starch. Okay. So I think you had better to take down this. How to prove that the above factors are required for the photosynthesis. Okay, so that's the topic. Next, before testing the requirements of the factors, we have to find a method to prove that photosynthesis has taken place. අපි දැනගන්න පුළුවන් වෙන්න ඕනේ මේක දුන්න photosynthesis වුණා. මේක දුන්නේ නැහැ photosynthesis වුණේ නැහැ. වුණාද නැද්ද බලාගන්න ක්‍රමයක් තියෙන්න ඕනේ. So take this down. After taking it down, so send me the message 1. කියද්දිම 1 කියලා message එකක් එව්වොත් 1 කියලා Chamidu, Tushani. So that means I ask you to keep your pen aside, but what you have done is, while I am explaining, you just take it down. Bad method of learning. Okay. I think time is enough. Good. Now, under this balanced chemical equation of photosynthesis, so this is initial product. Next, what happens after the polymerization? Starch is formed. Starch. What's this process? Polymerization. Polymerization. So what's the biological polymer? Starch is formed. එයාට නමකුත් කියනවා. So there are several polymers, biological polymers, and the carbohydrates. They are known to be polysaccharides. Grade 10, first lesson, polysaccharide. So this is plant polysaccharide. In the animal's body, another polysaccharide exists. What's that? That's glycogen. Okay. And in the plant body, structural, another polysaccharide exists. That is very good. Pume cellulose. Okay. So polymerization. So this is the main and initial product. Initial product. Polymerization happens later. But when we are going to test glucose, it has already been converted into starch. Or then if you test a starch for a plant leaf, we can observe starch has produced. Okay. If you test starch, putala starch test එකක් කළොත් plant leaf එකකට. Okay, so that's starch test. පුතා starch test එක positive වුණොත්, starch test positive, COVID positive වුණා වගේ නේද? Starch test positive වුණොත්, if starch test is positive, yes, you can see that glucose has been produced. Okay, so that means photosynthesis has taken place. If starch present, that means already photosynthesis has taken place. Therefore, we practice starch test for plant leaves to confirm that photosynthesis has taken place. Okay, take down under that. Although the initial product of the photosynthesis is glucose, although the initial product of the photosynthesis is glucose, large number of glucose molecules are polymerized to form starch. Next point. By the confirmation of the existence of starch, 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 starch පැවතීම තහවුරු කරලා confirm කරලා. By the confirmation of the existence of starch, we can prove that photosynthesis has taken place. That photosynthesis has taken place. Yes, that's what we do here. Okay. Next, starch test. So we are going to study in normal starch test. Next one, starch test for plant leaf. For a plant leaf. So these are the two titles we have to learn. Now keep your pens aside. So can you repeat the sentence? පුතා මේ මම sentence එක repeat කරනවට වඩා හොඳම වැඩි එහා පැත්තේ ගන්න වෙනවා YouTube file එක check it. අගෙන minimize කරලා තියාගන්න. මන්ට මේක දාපුවම මේක කරපු එක විනාඩියක් විතර late වෙලා තමයි ඒක යන්නේ. එතකොට you find it very easy to hear. Okay? පුතාලා දැනට space තියාගෙන යන්න බැරුව යන දේවල් බැරි වෙන එක හරියනවා ටික දවසක් යනකොට. දැන් උදාහරණ for an instance අපේ 6 වසරේ පන්තියන චූටි නංගීලා මල්ලිලාට ෆිසිකල් ක්ලාස් කරනකොට මට තියෙන ලොකුම ප්‍රශ්නේ තමයි එයාලගේ පෑන් වැටෙනවා. their pen is dropping down and පොත වැටෙනවා. ඉගෙන ගන්න පොත වැටෙන ළමයින්ට කවදාවත් ලකුණු 60කට වඩා එන්නේ නැහැ. හරි? ඒක නිකන් ආගමක් වගේ නේද? එහෙම වෙන්නෙම නැද්ද? ඔව් වෙන්නේ. ඒ වගේ තව මොනවද මං කියලා තියෙන ඔයාලට? ගෑනු ළමයින්ට. අම්මා තාත්තා එක්ක එකට එක කියනවා නම් especially for girls, තරහින් රවලා බලලා ගස්සලා හරි අර ඒ රියැක්ෂන් එක කළොත් කොච්චර මොලේ තියෙන කෙනෙකුට වුණත් ලකුණු එනවා හරි අඩුයි. හරි? මේක කියලා මොකද්ද අපි කරන්නේ? පෑන වැටෙන එක පෑන වැටුනොත් අහුලන්න දෙන්නේ නැහැ. 6 වසරේ පළවෙනි සතිය වෙන වැඩි. පෑන වැටුනොත් අහුලන්න දෙන්නේ නැහැ. ඒතකොට ලියන්නේ මොකක් හරි වෙන එකකින් ලියන්න ඕනේ පැන්සලෙන් හරි. ඒකත් වැටුනොත් කාගෙන් හරි අපි පොඩ්ඩක් බැනලා එහෙම දෙනවා. පොත වැටුනොත් වෙන කොලයක ලියන්න ඕනේ. So that single punishment what happens? සතියෙන් වැඩේ හරියනවා. නැත්තම් එක්කෙනෙක්ගේ පොත වැටෙනවා මුළු පන්තියම ඩිස්ටර්බ් වෙනවා. එයා ඒක අහුලනකම් අපි බලන් ඉන්නවා. ඒක අහුල්ලා ඉවරුණා විතරයි. What happens next? ඊළඟ එක්කෙනාගේ වැටෙනවා. ඉතින් මුළු පැය දෙක නැත්තම් පැය හතර පුරාම පෑන් පොත් අහුලා ඉන්නවා. ඔවා වෙනවා ඉස්කෝලේ. හරි? ඒවා ඉගෙන ගන්න තියෙන ඩිස්ටර්බ්. consciousness සිහි කල්පනාව දැන් මෙතන නම් එක්ස්කියුස් එකක් දෙන්න ඕන. මොකද්ද? පුතා සිග්නල් නැනේ සමහර අයට. සිග්නල් ප්‍රොබ්ලම්ස් there. කියන එක ඇහෙන්නේ නැති ටෙක්නිකල් ප්‍රොබ්ලම්ස් එනවා. ගෙවල් වල දැන් මයික් ඕෆ් කරන් හිටියට සමහර මේ වෙනකොට ටෙලිවිෂන් එක දාලා ඉතින් සද්දෙට තියෙනවා. ඒවත් මිනිස්සු කරගන්න ඕනේ. හරි? ඒකින්ද තමයි මම මේ අනිත් අතට YouTube ෆයිල් එකක් දාලා තියෙන්නේ. ඉස්සර නම් මේක කරපු ගමන්ම රෙකෝඩින් දාලා ආයෙත් ඉවරුණු ගමන් අප්ලෝඩ් කරනවා. So that's time consuming. හරි? හුඟක් විසීඩි මේ දවස්වල අපේ පුතාලා දුවලා O level ලියනවා. හරි? Okay, let's see. ඒකින්ද පුතේ sorry අපි මං අනිත් දවසේ and by using a dropper that reagent is applied. So what's that reagent? A few drops of a certain reagent is applied. Yes, very good. Iodine. Iodine කියලා විතරක් හරියන්නේ නැහැ පුතා. කියන්න පුළුවන් වෙන්න ඕනේ color එක. So what's the color of this iodine? Color of the iodine. Yes, good. Good. Brown, yellowish brown. Okay, you know that. Okay, so this is a sample of starch. Sample. What happens? So what's your observation? Yes, this is turned into dark blue or purple. Brown color iodine is converted into dark blue or purple. Okay, so that's the starch test. Draw a little sketch and take down these things. Okay, so if we put iodine on a certain substance, if it is converted into dark purple or dark blue color, that means that substance is starch. Okay, good. Okay, now I'm going to erase this. starch test for a plant leaf. පුතා දැන් a plant leaf figure, if we keep that in, not the plant leaf consists in pot plant. If we keep that in a place where there is enough sunlight for about several hours, 4 hours, if it produces starch, how do we identify that? How to prove that? පුතාලා මේකට අයඩින් බිඳුවක් දානවද? This experiment is in grade 9 science book. Though we apply this iodine on the plant leaf, it does not penetrate into the plant cell. Okay, so you know the layers of the plant leaf. So sometimes this epidermal cell layer, in addition to that, waxy cuticle layer exists. Wax layer exists. For instance, temple flower plant, temple tree, leaves. So this is a palisade mesophyll cell. Okay, imagine that. Or here, spongy mesophyll cells exist. Puta, where these starch exist, starch granules exist in a plant cell, where? In the chloroplast. Okay, look at this. So this is vacuole, this is the nucleus. Chloroplast can be seen like this. Chloroplast. I'll draw that somewhat magnified. So this is chloroplast. Okay, chloroplast. Double membranal organelle. Thylakoid, stack of thylakoid, we call that granum. Okay, and here's the matrix of the chloroplast. Here you can observe chlorophyll pigments on thylakoids. Yes, where this starch exists. Yes, produced starch can be seen in chloroplast as starch granules. Sometimes in the cytoplasm they exist. So whatever it is, whatever the place it exists inside the cell. එහෙනම් පුතේ අපි දාපු iodine. This is iodine, brown color. It just remains here. Iodine. This is iodine. Iodine drop. ඇඬු කඳුලින් ඉන්න ඇතුළට යන්න බැරුව. Okay, so that's iodine. Because plasma membrane, the living cells, plasma membrane, it does not allow any foreign chemical substance to go into the cell. Guard කෙනෙක් වගේ plasma membrane එක ඉන්නවා. It tells to this iodine, මෙන්න මෙහෙම කියනවා. Iodine කොයිද යන්නේ? So I want to meet starch because I want to prove the existence of starch. Then plasma membrane tells me මං ඉන්නකම් ඒව කරන්න හම්බ වෙන්නේ යන්න දෙන්නෙම නැහැ. So if you want to give the permission, so if you have to make this permeable, what gets permeable? Plasma membrane එක හරහා යන්න permission එක දෙනවා. So if you have to do so, here's the plasma membrane. So the starch first, it has to go through this epidermal cell layer. then it has to go through the cell wall of the palisade mesophyll. Cell wall can penetrate. If it penetrates the cell wall, the next thing it has to go across plasma membrane. Across plasma membrane. But it is impossible because this is a living cell. හුඟක් අය අපිට විවිධ යෝජනා ඉදිරිපත් කරනවා. New suggestions. Good. හිනහැනෙව කියන්න එපා කොල්ලෝ. rub the leaf. Leaf එක rub කරන්න. Good method. හරි. ජෝක් එකක් කළේ. හරි. ඒ පුතත් විහිළුවට කියන්න ඇත්තේ ඔක. Yes. Plasma membrane. Yes, how to make it permeable. So we have to make plasma membrane permeable. මෙතන තියෙන English ටික ගන්න. So yes, what we have to do? Yes, make plasma membrane permeable to iodine. Yes, how to make the plasma membrane and cell wall permeable to iodine. Because iodine tells like this, I want to go in to the cell, but this plasma membrane does not allow me to go. That means plasma membrane is not permeable to iodine. Yes, how to tell that? Plasma membrane is not permeable to iodine. Yes, how to take that permeability? මැරුණු දාට තමයි යන්න දෙන්නේ. එහෙම කියනවන්නේ. දුවේ ඔබට ඔය සම්බන්ධي කරගන්න මම දෙන්නේ මැරුණු දාට තමයි කියනවා. අම්මා නේද දුවට. දුව මොකද කියන්නේ? ඉතින් මැරියන්කෝ කිය. නේද අද කාලේම. So let's take down starch test for a plant leaf. Okay, right. Plasma membrane of a living cell. Plasma membrane of a living cell does not allow any foreign chemical substance to diffuse into the cytoplasm, across it, into the cytoplasm, across it. So that's the nature of normal plasma membrane of a living cell. Okay. Within bracket, take down. Plasma membrane is not permeable to foreign chemical substances. Next point. In the process of the conformation of starch theanogula, conformation of the existence of starch, in the process of the conformation of the existence of starch, iodine molecules iodine molecules have to diffuse into the cytoplasm, but plasma membrane of a living cell, is not permeable to iodine. Permeability occurs when only the cell or cell godak tissues, when the cell or the tissues are dead. මැරුණම තමයි permeability එක එන්නේ. නේ? permission එක ලැබෙන්නේ යන්න මැරුණන් පස්සේ. බට ඕක කරගන්නවා නම් පුතේ කරගන්න වෙන්නේ මේ අම්මගේ මලකඳ උඩින් තමයි කියනවානේ. So keep that in mind to keep this subject matter in your mind. Okay. Okay. So therefore, the first step 1, plant leaf, කඩාගත්තු plant leaf එක. පොල් කඩනවට මොකද්ද කියන්නේ? Plucking කියලා කියනවා. L අමතක කරන්න එපා. Don't forget L. Plucking. Okay. Yes. Plant leaf plucked. From the plant is boil in water. Now we know the reason behind that. So we have to make it permeable to iodine. මරන්න යන්නේ හරි. ඒක තමයි පළවෙනි කාරණය. So first step we practice is boil in water. හුඟක් අය එක කියලා තියෙනවා boil in water because we come across that in grade 9. පුතා හේතුව හුඟක් අය දැනන් ඉන්න ඇති. හරි? හුඟක් අය deeply දැනන් ඉන්න නැතුව ඇති. Or some of you know that, but some of you don't know it deeply. Okay? Boil in water. Kavisha, remove the waxy cuticle layer. Boil in alcohol. No, not that. Okay. So we have to learn this logically because exam questions are logical. If you don't learn this logically, you will give another answer what come to your mind. Okay. Next thing. Now you have boiled this using a test tube. So you have to draw it. Here's the test tube. And you put, no matter you can use a beaker. You can use a beaker as well. you can boil the plant leaf. Now the important thing, you have to select a plant that can easily be subjected to the starch test. මම කියනවා නාන ජෝර්ජ් ඔයාට පුතා starch test කරන්න suitable plant leaf එකක් අරන් එන්න. හරි? So George went to the school premises and searched a huge banana leaf. මෙයා උස්සගෙන එනවා කෙහෙල් අත්තක් බනාන කොළ. Sir, boil it in water කියලා. ඉතින් අපිට බැරල් එකක් දාගෙන මේක තම්බන්න වෙනවා. So therefore, we have to use a plant leaf that can easily be subjected to the starch test. Okay? Balsam. Right? දැන් කවිෂ පුතාට කියපුවම ගිහිල්ලා කියනවා එයා කෝමාරිකා. හරි? ඇලෝවෙරා ලීෆ් එකක් ගේනවා. වැඩක් නෑ. ඉතින් ඒක අපි suitable plant. Yes, balsam, bougainvillea leaf, manioc. So with a thin leaf blade can be used. Okay. So this is how we practice that. Boil in water. What's the purpose of this? Yes, to make the cell wall and plasma membrane permeable to iodine. Okay, take down the reason to make exam එකට අහනවා පුතා, why do we boil in water? To make the plasma membrane and cell wall permeable to iodine. Permeable to iodine. Hurry. Okay, now it's okay. පුතාලා print screen එකක් දාගන්න මේකේ. මේ තමයි ඔගොල්ලන්ගේ මතකයේ රැඳුණු චිත්‍ර රූපය. හරි? So that's your memory map. Okay? Memory developed image. So this iodine, it cannot move across this. Okay? Because of the existence of plasma membrane as is a living cell. So this is the cross section of plant leaf. Okay, waxy cuticle layer, epidermal cell layer. So this is palisade mesophyll, spongy mesophyll. So this is a chloroplast, and you can see chlorophyll pigments. So this red color one is starch granules. So this is your memory. මේ තමයි ඉගෙනගත්තු මතකය. print screen එකක් ගහගන්න, and you have to sketch that later. පස්සේ මේක අඳින්නත් වෙනවා. තේරුණාද? Okay, good. To make the plasma membrane and cell wall permeable to iodine, you boil it in water. Then වැඩි හරි. Now it's okay. මේ diagram එකේ ඇඳ ගන්න ඉඩ තියන්න. So you have to draw that. මේ test tube එක දානවා නම් ඒකත් අපිට පුළුවන්. So you can boil it by using a Bunsen burner. Okay. So here's the plant leaf. So two methods you can use. Both methods can be used. Okay, I'll erase that for your print screen. Okay, next. Now we have boiled it. We take it out from the boiling beaker or test tube. We take it out. This is the boiled plant leaf. So if you add few drops of iodine, what would happen? Now this cell wall and plasma membrane. So that's broken. So now it is permeable to iodine. පුතේ මේ බලන්න iodine ඇවිල්ලා starch granules එක්ක සම්බන්ධ වෙනවා. It combines with starch granules in the cytoplasm or chloroplast. Okay. Now, a color change occurs. Color change occurs. But because of the background green color, that color change cannot be clearly observed. Color change cannot be clearly observed because of that green color background. Okay. Due to the green color background, expected color change of iodine cannot be clearly observed. Okay. English. Perfectly. Okay. So you have to use English perfectly. Okay. So accurate English. පුතාලා නිකමට පොඩි වීඩියෝ එකක් ෆෝන් එකෙන් කරලා අප්ලෝඩ් කරන්න YouTube. අප්ලෝඩ් කරලා subtitles දාලා බලන්න. අපි හුඟක් වෙලාවට pronunciations වැරදි. අපේ English වැරදි. කම්පියුටර් වලට පිස්සු හැදෙනවා ඒවා දැක්කම. හරි? ඉතින් මේ තව softwares තියෙනවා. There are some other types of Android softwares. Your speaking style is belongs to which category? දැන් මම මේ උගන්න පාඩමක් රෙකෝඩ් කරලා දැම්මොත් එහෙම, so if I upload this recorded lesson, that particular software tell you this is a lecture, a kind of lecturing කියලා වැටෙනවා. දැන් අපේ පන්තිවල ඉන්න ගෑනු ළමෙක් සාමාන්‍ය විදිහට කතා කරනවා රෙකෝඩ් කරලා දැම්ම, that's kind of squirreling, රණ්ඩු වෙනවා, blaming එහෙම තමයි වැටෙන්නේ. So which category your spoken style belongs to, you can see. ඒකින්ද මේ language එක හොඳට හදාගන්න. So you are the most cleverest students out of all the students in your school, because English medium කියන challenge එක, that's a challenge. English medium students have to face two challenges. One is subject, another one is language. How these things tell in English. මේ කතා කරන දේවල් කොහොමද English වලින් කියන්නේ? නේද? How to tell that in English. Then මගේ lifestyle එක ගත්තොත් එහෙම කීයට පන්ති වුණත් නිදාගන්නේ 2ට විතර පාන්දර morning. Okay. So I have to get up early because I have to do some treatments to my father. So I have to get up. මේ නිදාගෙන ඉන්න බෑ. හිටියොත් එමේ යා ගෙවිලා යනවා. ඉතින් මේ time is limited. So ඔයාලට හුඟක් කාලය තියෙනවා මේක වැය කරලා ඉගෙන ගන්න. I never cram these lessons to teach you. මම ඔයාලට මේව උගන්වන්න කටපාඩම් කරන් එන්නේ නැහැ. එන්න බෑ. මට බැරිම වැඩ. ඒක හරි අමාරුයි වැඩක්. අමාරු නෙමෙයි, ඒක බැරි වැඩක් නේද? කරන්න බෑ. විශේෂයෙන්ම පුතාලා ඉස්කෝලේ කතාවක් පවත්වන, when you are making a speech, sometimes you cram it and go there. What happens? Stuck. කවිය මතක නැතුව ගියා නේද? ටිකරි නිසා හොඳට ගියා කියලා එහෙම මතක් කරන්න අපිට මාරිත් නෑනේ වෙලාවට කන්න. So my way is keep the theory in mind and allow it to flow. Subject matter එක ඔළුවට දාගන්නවා, allow it to flow. ගලාගෙන යන්න ඉඩ දෙනවා. So sometimes when I am remind or I am watching my own video myself, some of my friends and my sisters, brothers, so mother, so they are telling me this is the first time I see a person who smile with on lecturing. තමන් උගන්නපු පාඩමම hina වෙවි දන්නේම නැහැ. මට hina යනවා මම උගන්නනදී. මට තියෙන ප්‍රශ්නේ how I make this sentence instantly. So that's because of my English teachers. So I respect them. I always remember them. So one is Sir Ajit Tennakon, the Sarasari English Institute in Kuliyapitiya and Kurunegala. I don't know, yes, that renowned series Kuliyapitiya. So because of him, English is must. So පුතාලට English පුළුවන් නම් whatever the thought come to your mind, you have to convert that into English, not by your own way, by the standard way, not by English, but by English. Okay, so that's the way. So therefore, don't give up your English courses. You have to continue that. So this is a kind of English course. Okay. We apply iodine, it makes the color change, but expected color change of brown to dark blue, expected color change of brown to dark purple cannot be observed here. Color change happens, but expected color change of brown to dark blue cannot be observed due to the presence of green color background due to chlorophyll pigments. Okay, so next step, what we have to do is, okay, second step, step 2, step 2, boiling alcohol in a water bath. Yes, now you have to explain 2 reasons why it is boiled in alcohol, why it is boiled in water bath, why it is boiled in alcohol, and why it is boiled in alcohol, why it is boiled in a water bath. Yes, I like to talk to Sandali. Why do we boil that in alcohol? Okay. George, why do you boil it in alcohol?
To remove the waxy-cuticle layer.
No. Waxy-cuticle layer exists in the epidermis. Okay? That's not. Why do we boil it in alcohol in a water bath? 2 reasons we have to tell. I like to hear from Kavish.
So to remove the green color.
Green color. Okay, thank you, Puta. Green color. We are not going to remove that green color. Okay, green color එකක් remove කරන්න. So color pigment is to be removed. තේරෙනවද? Color and color pigment, 2 different things. Yes. මම හුඟක් කැමති ඔයාලත් එක්ක කතා කරන්න. අපි කවුද ඊළඟට ගන්නෙ? Yes. Tisun, what do you think?
To remove chlorophyll.
To remove chlorophyll pigments. හරි. මම time waste කරන්නේ ආයි ප්‍රශ්න අහලා. I think මම සතුටු වෙනවා ඔයගොල්ලන්ට මේ lesson එක නිවාඩුව කරපු එක ගැන. පුතා, we have to remove chlorophyll pigments. Okay? Therefore, we have to boil it in alcohol. Because in alcohol solutions, chlorophyll pigments dissolve. Chlorophyll pigments dissolve and washed away. Then my question, why it is boiled in alcohol in a water bath? What's the requirement of a water bath? Yes, Kavisha, very good. Chalana, good. Okay, හැමෝම දන්නවා ඒක. Alcohol is highly inflammable. If you directly heat it, its ignition temperature is very low. It starts to ignite. It starts to burn. එතකොට ටෙස්ට් ටියුබ් එක අතහැරලා පමෝත් මොකද වෙන්නේ? අපිට දුවන්න තමයි වෙන්නේ දාලා. So purpose of that is to remove chlorophyll pigment. Why? What's the reason? Due to that green color background, expected color change of iodine cannot be clearly observed. Due to the existence green color background, green color background created by chlorophyll okay, due to that අපේක්ෂිත වර්ණ විපර්යාසය expected color change of iodine. Expected color change of iodine. So use appositive phrase. Expected color change of iodine from brown to dark blue. Iodine from brown to dark blue. පැහැදිලිව නිරීක්ෂණය කළ නොහැක. Cannot be clearly observed. Cannot be clearly observed. Cannot be clearly observed. පුතා සාමාන්‍යයෙන් grade 11 වලදී බෝඩ් වල ලියන්නේ නැහැ චූටි ළමයිට තමයි ලියන්නේ. හරි? මම මේ මහන්සි නොබලා මේක ඉක්මනට ලියන්නේ. Some of you face problems due to signal and sometimes you cannot clearly hear what I explain. In that case, so this is helpful. Okay, third step, what we do, we take the plant leaf out and keep on a marble or white color background or tile. Then we add few drops of iodine and observe. Step 3. Take the plant leaf out and keep it on a white surface. ඔය ටයිල් කෑල්ලක් marble sheet එකක් උඩ ඔයගොල්ලන්ට තියන්න පුළුවන්. Piece of white paper can also be used. Keep it on a white surface and add few drops of iodine few drops of iodine and observe. Okay. Observation 1. Conclusion 1. Observation 2. Conclusion 2. Observation 1. Expected color change is observed. What's the expected color change? Expected color change of iodine is observed. මොකද්ද පුතේ? Dark brown to brown to dark blue or brown to dark purple. Expected color change of iodine is observed. Then your conclusion? Starch has produced. ඕකනේ directly කියන්න පුළුවන්. That's the direct conclusion. Starch has been produced. Iodine blue වුණා කියන්නේ starch තිබ්බා කියන්නේ. Okay, then the second, you can conclude that starch has produced, but starch is produced by the photosynthesis. Your final conclusion, photosynthesis has taken place. Photosynthesis has taken place. Yes. On the other hand, you may observe no expected color change. No expected color change of iodine from brown to dark blue. So that means conclusion. Photosynthesis. මේ කියන්නේ අපි කියන්නේ ඉස්සරලාම කියන්නේ no production of starch. No starch produced. That means photosynthesis has not been taken place, or photosynthesis has not occurred. So this is the complete starch test for a plant leaf. Good. Okay, next, experiment 1. Experiment 1. Yes, what that first experiment? To prove that to prove that sunlight is required for the photosynthesis. Yes, in this case, we have to give sunlight and check whether the photosynthesis has taken place. Okay, next, we don't give it and we control the controlling factor sunlight, and we do not supply that, and we observe whether the photosynthesis has taken place or not. Okay, in that case, we have to use earlier described starch test for a plant leaf. Okay, දැන් පුතාලට මහන්සි ටිකක් පෑන තියන්න පැත්තකින් අන්තිම විනාඩි 5 rap song එකක් වගේ මම experiment එක explain කරනවා. Try to understand. Okay, here you have to use a pot plant. Okay, so you have to use a pot plant. Why do you use a pot plant? So we have to move it here and there. Okay, sometimes we have to keep that in a place where there is enough sunlight. Okay, so it's a pot plant. We have to supply sunlight as well as we have to control the sunlight. So we have to use 2 opaque black strips of papers, and we both sides cover the plant leaf with the portion of the plant leaf is covered with 2 black opaque strips of papers like this. Then sunlight is absent to this area, upper surface and lower surface. Okay. When selecting this plant, first we have to keep it 48 hours in the darkness. 48 hours in the darkness. What's the reason behind that? Because already produced starch exists inside this covered area. Already produced starch exists. Okay, if we keep this plant without keeping it 48 hours in the darkness, we keep it in a place where there is good sunlight. Okay, we keep this plant. Now it is exposed into sunlight. We keep that around 4 hours. Now time is enough for the photosynthesis. Now what we can expect? Starch has been produced. Now we send this to starch test, boil in water. Okay, boil in alcohol in a water bath. Boil in alcohol in a water bath. Okay, කලින් පුතා experiment එකේ අපි මේ රූප සටහන ඇන්දේ නැහැ. We did not draw this. Okay, when you rewrite that, you have to draw it. No matter you have learned that in grade 9, if you draw it, I like that. Okay, so you know the reason. Boiling in alcohol in a water bath, you know both reason. So this is starch test, and finally you test it for iodine. So in your tute, you can see this experiment. Then expected color change happens. Okay, I'll draw the result here. This expected color change can be observed both in covered area as well as open area. මේ හැම තැනම පුතා expected color change කියනවා. Dark blue color can be observed. Why? In this covered area, the already produced starch exists, therefore expected color change use. Here, already produced starch and new recently produced starch exist. දෙකම තියෙනවා මේ පැත්තේ. හොඳට blue වෙනවා. ඉතින් අපිට ඔප්පු කරන්න පුළුවන්ද? Can we prove the experiment? Requirement of sunlight? No. Therefore, we have to keep it 48 hours in the darkness to remove already produced starch from the plant leaf. Because at the night, this already produced starch is converted into sucrose and dissolved in water through the phloem tissue. Already produced starch is removed from the plant leaf and stored in yams, fruits. Okay, so that's the reason. පුතේ පැය 48ක් අඳුරු තියන්න ඕනේ. ඇයි? දැනටමත් හදපු ස්ටාර්ච් මෙතන තියෙනවා. හරි? ඉතින් කළු පාට කොලයකින් වහපුවම මෙතෙන්ට sunlight වැටෙන්නේ. So in this area is exposed to sunlight. When we allow it to do photosynthesis by keeping that in 4 hours in exposing to sunlight, හොඳට ඉරෙලිය වැටෙන තැනක පැය 4ක් තියලා අපි ගත්තාම කොලයකින් වහලා තිබ්බට වැඩක් නැහැ පුතා. ඇයි? දැනටමත් හදපු ස්ටාර්ච් මෙතන තිබ්බා. මේක බලන් ඉන්න කෙනෙක් කියන්න පුළුවන් මේ බොරු වැඩ කරන්න එපා. ඔය සන්ලයිට් ඕනේ නෑ. බලන්නකෝ ඔයා වහලා තිබ්බ තැන කලර් චේන්ජ් එක වෙලානේ. ඒ කියන්නේ ලයිට් නැතත් photosynthesis වෙලානේ කියලා කෙනෙක් කියන්න පුළුවන්. එහෙනම් මෝඩයෝ ඉන්නවනේ සමාජයේ. ඒ මිනිස්සුන්ට අවස්ථාව දෙන්නේ නැතුව as scientists, what we have to do, we keep that in 48 hours in the darkness, then already produced starch is removed from the plant leaf. තේරුණා නේද? ඉතින් පුතා අපි මේ පන්තිය අර කලින් කිව්වා වගේ පැය 4ක් කරනවා සාමාන්‍යයෙන්. අද තියෙන ප්‍රශ්නේ 10ට පටන් ගන්න ඕනේ පේපර් ක්ලාස් එක grade 11 මේ පාර විභාග ලියන අයියලා අක්කලාට. ඇත්තටම අද කරන්නේ all the chemistry සියලුම රසායන විද්‍යාව grade 10 වල ඉගෙනගත්තු දේවල් සහ grade 11 physics. ඕගොල්ලන්ට grade 10 chemistry අප්සෙට් ගිහලා තිබ්බනම් ඒ ඔක්කොම අද උගන්වනවා මේ විභාග ලියන්න එන අයියා අක්කලාට 10ට. මට මේ පන්තිය තව පැයක් කළොත් if I continue this class another 1 hour, so it is difficult for me to stand around 6 hours at straight. පැය 6ක් එක දිගට හිටගෙන ඉන්න අමාරුයි. හැබැයි මම කරලා තියෙනවා. ඊට වැඩිය හවස 6 ඉඳන් පහුවදා 6 වෙනකන් හිටගෙන ඉඳලා තියෙනවා මම. හරි? ඒක මට ගැටලුවක් නෙවෙයි. මට මේ උගන්වද්දි ටයිම් යනවා තේරෙන්නේ නැහැ. නමුත් ඒක හොඳ නෑ. ඊයේත් මම මේ වගේ නිදාගෙන නැතු නිසා මට දැනටත් ඔළුව කදේ. හරි? ඔයාලට පෙවුන්නේ නැහැ නේද? හරි? ඒක මට දැනෙනවා. But pain cannot stop me. එහෙනම් පුතේ අදට goodbye to all of you. So revise these things. Look this. You have to watch this video again and again. Okay? If there's any doubt, send me. Okay? So now I am going to switch on your microphones. I like to hear your voice. Goodbye. Thank you, Aibuan. So that's the energy. Okay. So have a nice
Good
night.
sir. Goodbye, sir.
Goodbye to all of you.
Bye, sir.
Good night,
Goodbye,
sir.
sir.
Thank you, sir.
sir.
Goodbye, sir.
Thank you.
Goodbye.
පුතාලගේ ක්ලාස් එක එන්න බලන්න 10ට. හරි grade